Many G protein coupled receptors have a large extracellular ligand binding domain. When the appropriate protein ligand binds to this domain, the receptor undergoes a conformational change that is transmitted to its cytosolic regions. This activates a trimeric GTP binding protein. This protein is comprised of 3 subunits: 1 alpha, 1 beta, and 1 gamma particle. From this point forward, we will refer to this intact triunit as a G protein. Alpha and gamma subunits possess covalently attached bonds to the lipid tails of the phospholipid bilayer. This serves as an anchor for stabilization in the plasma membrane. In the absence of a signal, the alpha subunit has a GDP bound. For simplicity going forward, the 2 black bars shown here will represent the phospholipid bilayer. In the presence of a signal, binding of the receptor and G protein causes a conformational shape change in the alpha subunit of the G protein. Because of this shape change, GDP will dissociate, allowing for the GTP present in the cytosol to replace the GDP in the alpha subunit. GTP binding now induces further shape change in the alpha subunit of the G protein, thereby activating not only the alpha unit but also the gamma-beta complex. It is significant to mention that the activated beta-gamma complex can go on to regulate its own independent processes. However, for this video, we are primarily going to focus on the effects induced by the activated, GTP-bound alpha subunit. This unit can now regulate the activity of target proteins bound in the membrane by conferring a conformational shape change. It is important to note that there are many types of target proteins. However, for the purpose of this demonstration, the target protein known as adenylyl cyclase will be the main focus. The activated adenylyl cyclase relays the signal downstream, initiating a signal cascade. ATP, which is always present in the cell, is used for the synthesis of cyclic AMP. This conversion is specifically catalyzed by an activated adenylyl cyclase. Cyclic AMP is specifically referred to as a secondary messenger, the first messengers being the extracellular signals. Cyclic AMP then in turn activates a cyclic AMP dependent protein kinase, otherwise known as pKa. Activated pKa can continue on via 2 pathways. The first pathway that will be discussed results in glycogen breakdown. pKa will phosphorylate and activate an enzyme called phosphorylase kinase. Phosphorylase kinase then in turn activates glycogen phosphorylase, the enzyme that breaks down glycogen. Recognize that every phosphorylation step is occurring by converting ATP to ADP. As stated previously, pKa can continue by an alternative route. In this route, the active pKa migrates into the nucleus and phosphorylates specific transcription regulators. Once phosphorylated, these proteins stimulate the transcription of a whole set of target genes.